It's just us today. No Dariia today, unfortunately. She's okay though.
Let's see. Turn the volume here. All right.
Yeah.
Wait for people to
Checking
join.
on the trip. Yeah, my
Pumpkin seed.
see it on Facebook. Let's see.
Yeah,
It's
usually
supposed
it takes a second to kick in for all the different platforms. So
Yeah, I tried to link it directly to my Patreon today, so I'm gonna see. But it's weird because you have to schedule it, right? Like you can't just be like activate because it doesn't go through Restream. So mine should kick on and Patreon in
I
a
think that
couple
about Patreon. They don't have like a live stream thing for Patreon.
I mean, they kind of do, but you just sort of like link your YouTube live and then schedule a post to go on. But like you have to do it at least 5 minutes in the future. So, so I guess they're like, okay, so you schedule it, then you schedule your live stream, then you sit down and the live stream starts and it posts at the same time and everything starts. So
yeah, but we'll we got what's up, Joey? Yeah, we got the live stream working on the CDC website. Unfortunately, I think people who are using the CDC website don't have chat.
Yeah,
you want to chat, you have to find the stream. The problem is like Facebook doesn't. I don't think they have like a hard link for live streams. And right. Twitter, I don't know. And Twitch, so I've been sharing the Twitch live stream link. Mine is twitch.com/dubiousbattle. Dubious Battle, which is like a reference to John Steinbeck book.
Yeah. It looks like it's working on all the CDC
book people out there. Yeah. Anyway.
But yeah. Should be on the site now too. Okay, good. Good.
All right. So today we don't have Dariia. She couldn't make it, but it's all good. Last week we performed our experiment to extract and isolate our stem cells from our fat tissue.
Yep. That's pretty exciting.
And so that's what we're going to talk about today. We'll get more information up on the CDC website, the-cdc.com. So if you want to check out protocols and stuff that we did, just be on the lookout for that. Otherwise, today we're just going to, share what we did and the experience and the results and stuff like that. Maybe some pictures and stuff, and we'll see. We'll see what I can find on my laptop.
Yeah, and there's what do you think that other video will be ready?
Yeah, I mean, we'll have video and we'll have more stuff on the CDC website soon. But yeah. So if you haven't been following along, what happened was we decided to try and isolate stem cells from our fat tissue. This is a fairly common procedure, especially done at like stem cell clinics and shit like that. Except usually what they do is they'll do like liposuction, suck out a bunch of fat and blood and other shit, and then they'll centrifuge that to get, quote unquote stem cells, and then inject it back into people, which I don't know, questionable. I'm not gonna say that you should do that stuff, but like, does it actually do anything? I don't know, questionable science and medicine. But we wanted to isolate our own stem cells and grow them up. The process was a lot simpler than we anticipated, I think.
Yeah, especially for me. I mean, I was like, I had kind of considered the idea vaguely in the past, but I was always like, nah, that's going to be way too involved. But yeah, it's kind of surprising.
Yeah, because, well, originally we wanted to isolate our stem cells from our blood because we wanted something that you didn't have to like stab yourself for. But that process seems a lot more complicated. I don't know. It's something I want to revisit with that and actually see how complicated that process is.
Yeah,
Because looking at this process for isolating stem cells from fat, it wasn't as complicated as like I anticipated.
yeah,
Um, Basically, what we did was we took a needle and a syringe and stuck it, beneath the skin in our, belly, and pulled back on the syringe, the plunger, and like sucked in some fat and tissue and whatever else was underneath the skin, and took that and did a collagenase digest, which I don't think you need.
yeah.
I'm going to have to ask Dariia about that. Maybe I could text her right now, but did a collagenase digest. Um, let's see, Dariia. And then just put it in media, normal media, which was like L15. We use L15. Most people use DMEM and CO2 incubator, but we're way too
We're
smart to
too
know
cool for
that.
that.
We just use L15 in a normal incubator without CO2, and everything works fine.
Yeah, uh,
I just don't get it.
yeah,
That's one of the things that's going to baffle me. Like 20 years from now, finally people are going to be like, oh, yeah, why do we use CO2? And I'm going to be like, that's what I've been saying all along.
I know. Yeah, it is this weird, like, archaic thing that's just like still around, and everybody thinks you need it.
Yeah. But I don't think you need the collagenase digest step. Um, like, I think you can just probably put the tissue directly into L15. Um, and you know, FBS, and it should work just fine. But we haven't tested
Yeah,
that yet. But like, there shouldn't really be any difference. What collagenase does is it just breaks down the collagen, so the cells separate better, and so the cells, you get they're able to proliferate and grow. You can imagine if a cell is attached to a bunch of other cells, its chances of like landing on the flask and sticking and then growing and replicating are very small. Or if it's like inside a clump, like it's not going to get any media. And so collagenase helps. But like we got a lot of cells. We
yeah,
got a lot.
for a pretty quick process, like it really
Yeah.
wasn't. It wasn't as scary as I thought it would be. Like I really expected it to like be a lot harder. Like I don't know. Part of me feels like you'd stick a needle in, you pull back, and just nothing would happen. Like I feel like it should be better put together than that. But yeah, I mean, you had to work it a little bit, but cells just flow right in.
Yeah. Let me see. I think I might have video of the cells. Um,
Oh, can you share your screen?
yeah, let me find.
Yeah.
I think I have like a picture of the cells, and yeah. So we started. Let's show the cells here. Let me share my screen. Um,
Let's see.
Screen share. Entire screen share.
There we go. Oh, we're
How
still
do I
here.
make it full screen without the chat?
We're in a mirror hallway now.
This is great. Come on.
One of these options has got to be the one.
All right. Well, we'll just do this. I
I
don't
think
know.
you'll just have to click away to the other thing. Oh my
yeah,
gosh, you have even more tabs than I do.
All right. So here is a little video of the flask and the cells. So you can see all these little weird shaped things are stem cells, or what we think are stem cells. We're working on inducing them now just to verify they're stem cells. They do look a lot different than the fibroblasts that we've grown in Purify, isolated and grown before. And so it's very promising, and it's hard when you look online at like stem cell pictures, like everybody's stem cells looks different. So these are mesenchymal stem cells, or they should be mesenchymal stem cells. And I can't imagine what other cell type these would be.
Right.
And so yeah, I can't imagine what other cell type these would be. And so, I think I would say we're pretty sure they're stem cells, but now we're working on inducing them into other tissue types to just verify if they are stem cells. And that takes a little bit of time, but that's also really cool that now we can try to grow different tissues with our own stem cells.
Yeah, and it's a surprisingly broad number of tissues for the same stem cell. Like the mesenchymal stem cells are pretty potent. Like I'm kind of surprised how many different tissue types you can make.
Yeah. It's like what neuronal tissue, muscle tissue, bone, like cartilage, or like
Tendon and stuff.
Tendon and cartilage stuff. What do they call that? Is that like soft, like they call it chondrocytes?
Yeah.
I mean, is there like a word for that? And then, is there anything else I'm missing?
Probably, but yeah, they just become like a lot of things. It's interesting that all those tissues have like a common like origin. I mean, obviously, all your tissues have a common origin. You go far enough, but you just kind of figure. I don't know. If I had to draw it in my head, it probably would have connected differently. Nature's just unpredictable.
Yeah, it's funny because like before I do this, I never realized why people use fat, but it totally makes sense because there's like no other cells in there, right? Like the fat tissue, you shouldn't get any other cells besides stem cells, which make fat cells and fat cells. And fat cells, well, number one, they float, so they're easy to separate,
right?
And number two, like they don't replicate, and so they won't like stick to the flask or anything like that. So realistically, the only tissue you should be able to get from your fat is stem cells. There's no other tissue that you can harvest like that and get like just stem cells.
Yeah, I mean, you might get like a few white blood cells or something in there if you get a lot of blood, but like, who cares? That's not going to really affect anything. And like,
so it makes, it's crazy. I never thought of that, but I'm like, that's kind of smart,
Yeah. Somebody asked about scalability.
What do you think
I don't know what the largest scale we want to grow is. I mean, like maybe one of those big flasks I could see if we were just getting crazy with something, or if it works really cool.
I would say like, what's the point?
Yeah.
Like if you're talking about like re-injecting them back into ourselves to give us stem cells, I don't really believe in that type of stuff necessarily. Like you have to understand a stem cell is just a cell that hasn't yet reached its cell fate, like it hasn't reached its end lineage. So like if you put stem cells, if you inject them into your muscle or something like that, like the majority of them are probably just going to die or go into your bloodstream or some shit like that. Some of them might turn into muscle cells. Some of them might turn into fat or skin, or generally stem cells will turn into whatever cell that they're like next to, or whatever
Yeah.
cells like secreting the factors that convince them to turn into something. Um, and so, but like you're not gonna cure yourself of anything really. You're not gonna like change anything in your body or, you know. And so it's like, yeah, stem cell, like growing
Yeah,
stem cells in that or something isn't like, I don't
and
know.
as easy as these were to collect, it's a good demonstration of how common they are already. And like, yeah,
Yeah,
we're younger, so I mean, I don't know, maybe it changed, but so stem cell trans. So I think you mean bone marrow transplant, which does have stem cells, but then you're effectively like you're talking about the Berlin patient. So man cured of HIV following breakthrough stem cell function. But that's the Berlin patient. Basically, they took somebody who already
yeah,
had
that's not stem cells.
system.
Like I don't, whatever. Like it's
Yeah,
not stem cells. Like you're talking about transplanting an organ, not stem cells. That's what they
exactly.
did, and like stem cells don't like. There's been so much touted about stem cells as like this curative agent, but like in reality, it's all anecdotal. It's like athletes go and get their stem cells purified and re-injected into their knee because their knee hurts, and it like, ooh, it feels better. But it's also like that doesn't really make any sense.
Yeah,
I mean, I
yeah, and,
guess maybe something. It's probably like all the lidocaine they gave for all the injections that like it reduced the inflammation or something,
yeah.
rather
Or it
than
could
like
just be like
cells doing
adding
anything.
soft tissue. Like I've seen like collagen injections in knees and stuff, and it's just protein and it's just squishy stuff in there where bones are hitting bones normally. Because your
Yeah,
knees are worn out. But yeah, as far
yeah,
as could you give yourself a better immune system? Like better immune system, I don't know what that means, but like people have done like basically immune system transplants for immunocompromised people, but just a bone marrow transplant. Like that's not an uncommon thing.
yeah. I mean, stem cells like you're. The thing is like, do stem cells do stuff? Yes. Are stem cells some curative thing that cures a disease and can do everything? No,
Yeah.
no.
You got to
Stem cells.
remember, yeah, like just because like an athlete does it doesn't necessarily mean there's anything to it. Athletes also do like cupping and you know all sorts of crazy stuff, right? So I mean, like, who knows if any of this stuff? Like just because
Yeah,
an
I
Olympian,
mean,
doesn't mean that's the secret.
stem cell injections and stuff like that are just very, yeah, very suspect. Um, I'm not saying that like they can't ever do anything or they don't do anything. Like they do, but the thing is they don't like cure disease. What they do is they turn into cell types
Yeah,
that are they're around,
yeah,
you know. And what does that mean? I don't know. Like I don't really know. Like can it heal stuff? I don't know. Maybe if you had like, heart muscle tissue that died, stem cells could come in and become new tissue. Maybe in cases like that, you could see stuff. I don't know, like where there's actual tissue damage.
yeah,
These cells are replacing other dead cells or old cells or something like that. But like, is that possible? Does that work? I don't know. Like, you're basically just injecting something in your body and hoping for the best. You can't really
yeah.
control
And
cells.
especially if they're not like modified in any way, you're just injecting something in there that's already there. You
Yeah,
know,
they're already in your fat tissue,
yeah.
so
And
it's
I'm sure they migrate around a certain amount.
yeah,
I mean, mesenchymal stem cells are in bone marrow, they're in fat, they're probably in everything.
yeah. I think they're probably in every tissue. They're just easiest to get from fat, because like definitely in my fibroblast cultures, I've seen weird looking cells that definitely didn't look fibroblasty.
Yeah.
Are they? I mean, here's the problem: is like most cells look the same,
Yeah.
and so like, or they're just like cells can be the morphology can be very flexible, and so like identifying a cell type just through morphology can be difficult, and so stem cells, identifying a stem cell just by look is hard. Um, Stain them, I mean, that doesn't, you can't just stain stem cells. Like you can talk, are you talking about using antibodies to try to find certain stem cell? Sure, you can try and do that. But that's fairly complicated, especially in a living culture that you want to use. Um, it's not something that you can just easily do or want to do.
Yeah. Not to mention, the
That doesn't even make sense. Just are you just saying random words? Sounds like it. No response from Daria. I don't know if her cells are alive or if she's even checked yet. But I
yeah.
hope I can't imagine that they aren't.
Yeah. Yeah. I mean, as many as I got and you got from just ours, I can't imagine. I feel like an explant would have, you know, good odds.
Yeah, I agree. Like, yeah, there was a surprising number of cells in there. Um, and so generally, what they do is they, take the tissue, destroy the red blood cells using ammonium chloride, which we didn't do. We didn't think it would be necessary, and it totally wasn't necessary. Um, and then collagenase digest, and then just put in the flask.
Yeah.
Um, Obviously, we washed it to remove collagenase and other stuff, but like those were the major steps. And yeah, there were a good number of cells, and like
Yeah. We.
yeah, considering the amount of tissue I got, it was pretty small amount of tissue, but pretty a
Yeah.
large amount of stem cells.
I got a larger amount, but then when I was pouring out the media from my after I centrifuged it down into a pellet, I poured it off, resuspended it, poured it off, centrifuged again, and I was pouring it out in the sort of bleach water, and I was thinking, okay, there's this little drip hanging on. I'm just going to shake it loose, and about half my pellet just went floop into the bleach, and so about half my cells are just insta dead. And so I still got plenty of cells, even though half the stuff I collected was gone.
Yeah, that's
That's a surprising amount available.
yeah, yeah. And I put mine in like 2 wells on a 12 well plate, and then I removed the media and I put it in another 2 wells on a 12 well plate, and then I removed that media and threw it out. And like the second set of wells also grew a ton of stem cells, so like
Nice.
there was plenty in there.
So
Um,
you
and
waited two
and
days before you
initially,
published your media?
yeah, I let it sit 2 days and then I changed my media. Yeah, after 48 hours.
Yeah, that's what I did. I think the yeah, mine I had a bigger pallet, even though I lost half of them, and so I took the uh, I made 4 wells at the beginning, and I've just been changing media. So I probably threw away quite a bit of cells since I didn't, sort of
I was
re.
also surprised that these cells are happy in just L15 with 10% FBS,
Yeah,
like just happy and fine.
yeah, and they're
Grow
growing.
great.
Yeah,
Yeah, growing great, and you don't need any weird or different media or any special factors or anything like that. Um, yeah, so it's just like, but I think I mean, one of the big things is opening up, um, the creating more opportunities for people to do experiments and research
yeah, yeah,
because like that's the thing with these, primary tissue culture and isolating stem cells and stuff like that. It's like I think maybe the next project should be is like creating, you know, uh, induced pluripotent stem cells from our own cells.
Yeah. And I wonder if it's easier to make, like maybe you need fewer factors if you start with mesenchymal stem cells or something.
Maybe, but yeah, I mean,
And
it's usually a pretty standard procedure nowadays where you can just like
yeah,
buy a kit online and just like transform your cells and get, induced pluripotent stem cells from your cells, which is like, yeah. Yeah, the stem cells, like we just talked about, surprisingly, they just grow off a normal media that you grow like any generic cell type in. Um, yeah, like we didn't even heat, sometimes people heat and activate the FBS or like these companies, they sell, like mesenchymal stem cell media
yeah,
and like it's bullshit.
yeah.
Um,
This stuff, it's like Kool-Aid for cells.
yeah, but it's great. Like the reason we're doing these experiments is to show what's possible, to differentiate the cells and see what cool stuff we can make and do with them. Um, yeah, it is, Robin. It is the same media we use for HEK cells and other cells. It's just FBS, 10% FBS and L15. I think we use L15 F12, but the only reason we use L15 F12 is because it has a slightly better buffering capacity, I think, than just L15 by itself. It's just half L15, half F12 media. Um, but that's pretty much it. Uh, yeah, but yeah.
Yeah,
Um,
yeah. The FBS is still the expensive part. Can stem cells be grown without embryo? Yeah, that's what we're doing. It's like we're growing our own stem cells from us, adult people.
yeah, but yeah, I think creating. I mean, the thing about creating induced pluripotent stem cells is like literally, um, you can just buy a kit nowadays.
Yeah, it is kind of crazy because that shit was like,
Should we make like that? Might be an interesting idea. Is to make like open source plasmids for creating induced pluripotent stem cells.
yeah,
Because I think you need four factors, and I think what you can do is you can do like four plasmids or two or one plasmid using the 2FA or whatever cleavage site.
yeah. Self-cleaving
Yeah,
peptides. Yeah.
yeah,
I wonder if you could make a
um,
single plasmid system.
yeah. I think people have and do, right? It's huge, um, but like, right, because it's got four genes on it. So you're talking something that's like, oh, I forget how big it is, but something like 12K or 15K or
Yeah.
something like that, as opposed to like, 4K, 5K. So obviously it affects transformation efficiency, but like I've seen companies that sell that. That might be an interesting thing to do is create, a single plasmid, um, open
I mean,
source it so people can create induced pluripotent stem cells, you
yeah.
know.
It would be interesting. I know there'd be a lot of people interested in doing it. You know?
Yeah, yeah. I mean stem cells in general, you know. I think it's gonna be I think it's going to be really cool, like to eventually start selling kits that allow people to work with stem cells. I think that's our goal, is to release a stem cell kit pretty soon that allows people to grow and induce stem cells.
Yeah.
How fucking cool is that? I mean, I don't know.
Yeah. Well, funny thing is, what two populations of stem cells. So we'll have to see like which one. I don't know. I imagine they'll do exactly the same, but yeah, maybe we'll have a contest. See You can vote whether you want my stem cells or Joe's stem cells.
Oh, maybe, but I think that's weird. I think we just send them cells anonymously.
Yeah.
There's enough creepy people out there that.
I know, right? That's exactly what I was thinking. People can do weird stuff. Your emails will get even worse.
The thing is that the stem cells don't really last very long. So, right?
Yeah.
So you'd have to like grow them up, split them, send them out, and then, I mean, I guess we could freeze a bunch, but I imagine even if the stem cells we have right now, even if we grow them up, after 2 passages and freeze them down, it's still not going to be a ton of cells,
Right.
not enough to like. So I imagine that like.
You'll need several passages once you get the kit to do
Yeah.
the production and stuff. Yeah.
Yeah. So I imagine that like usually cells will, will be more than once that we'll need to get something, but it's not that bad. So it's
Go fishing.
yeah.
Yeah. Ugh.
But I'm super
Selecting
interested.
microbiome flush stem cell growth.
It's a really hard one because growing any sort of bacteria in culture with cells is
Yeah.
complicated. So if you're trying to do experiments with something like that, it's really not easy experiments to do. If you're talking about in real life, like you want to change your microbiome to foster stem cell growth, I don't really.
Yeah, that would be a difficult one to try and evaluate success on.
Yeah.
It's like, how do you tell?
And like, why would you want stem cell growth? Right? That's the thing we've talked about, and I'm sure we'll just repeat ourselves over and over again. Like, stem cells aren't a magic thing.
Yeah.
Like, most of your stem cells are just making cells that can't replicate.
Yeah.
Right? So,
Yeah.
like, your stem cells are making like fat tissue or bone or things that can't replicate. Right?
Epithelial cells and yeah.
Yeah. Or things that die really fast and continuous. Like, your body's full of stem cells, and like helping stem cells doesn't really like. The thing is, there's a Roundup, Roundup for like weed killers. They generally use this compound 2,4-D and like 2 number 4 and then letter D.
Yeah.
And what it does is it causes the cells to revert to stem cells. So like you use this compound 2,4-D when you're trying to grow calluses. And like all the cells turn to stem cells and the plant just dies because it like freaks out and can't do it. So you have to think in your body, if you just like want all these stem cells, it's not good. Like it's not necessarily a good thing, right? Like stem cells are not a thing that like have eternal benefit and eternal goodness in you.
Yeah, I mean, yeah, if you just turned yourself into a bunch of stem cells, you'd just be a blob of undifferentiated cell mass and not a person anymore. You'd just be an ooze.
Yeah, you don't want like making your stem cells replicate more or something in your body is.
Yeah, I think so. I think a lot of this comes from a lot of the people that are selling stem cell therapy as a miracle cure for everything, or who are just like, selling various probably supplements. All the worst
Don't
people.
get me wrong. If I had like something and there was no other treatment and I thought stem cells could help, maybe like I would for sure try it.
Sure.
Like they do stuff. They do, especially when there's like tissue damage or something. Can they cure cancer or cure other shit? No, like that doesn't really make any sense.
No.
But they do stuff.
I could see it like if you had like a genetic disease like that German boy whose like skin was falling off. Those weren't specifically skin cells or skin, Yeah. Well, anyway, he had genetic disease. He had like really bad skin connectivity, so they like genetically modified skin cells, grew them up in big grafts, and started patching them on there, and eventually it colonized. But like, there's a really strong selective advantage to the modified skin that's like functional versus the skin he was born with, which isn't functional. So I mean, I can see stuff like that. Like if you've got like
And
some,
wound healing. We were talking about that, the paper we were talking about previously, wound healing, like stem cells help in wound healing. If you have like a wound or something like that and taking your own stem cells and seeding them, which might be something fun to try and interesting. You just gotta wound yourself.
all I got to do is wait. I'll eventually wound myself. I'll be doing some yard work. I'll get a big
The thing
gag.
is you need like two equally,
Oh yeah, you need like identical, so you have a control. Yeah. That's probably
you
the
get like scratched by a cat or something, and then you got like 4 wounds, and you can choose like 2 for stem cells and 2 the control.
That's not going to work because I don't have a cat, and your cat's too nice. He would never scratch anyone.
James does, but only like usually little kids who like pull his hair.
Yeah.
Yeah. Yeah. My kids always get scratched.
Okay. Yeah, and I could see maybe like a stem cell transplant from another person who doesn't have the same genetic disease you have or something doing some good.
Oh yeah. But that might be, yeah. I mean, the good thing about your own stem cells is the compatibility.
Yeah, well, that's the nice thing about bone marrow transplants is because you just take that person's immune system when you're done.
Yeah. Don't they like irradiate you to like kill all your bone marrow?
Yeah.
And then they,
When I was born, or before I was born, that was the plan, was for me to be a bone marrow donor for my brother and sister.
Do you think the stem cell application will reduce scarring? Yeah, I think so.
Be interesting. But yeah, so I mean, it's like they wanted they were both sick, had immune system disease, and so they figured a bone marrow transplant was the only way, and they were hoping that when I came along, because I was the youngest, that I'd be a compatible donor and be able to donate to both of them. But then it turned out I had the same problem, so it wasn't very useful. But luckily, shortly after that, my brother outgrew the problem. His immune system kicked on, and then my sisters did, and then mine did. But yeah, so I mean, bone marrow transplant's pretty old. I mean, it's at least like 30-something years old.
Yeah, let's talk about these stem cells, with wound and scarring and stuff like that. That's a cool one.
Scarring is interesting because, like, I mean, when you get cut, don't cells proliferate? Like a lot of fibroblasts and stuff proliferate in the area and make a bunch of collagen. And isn't that what scar tissue is? So, like, would the stem cells necessarily do a better job?
I am pretty sure I've read papers on this.
Interesting.
Right? The thing about these experiments though is really, it would be interesting to see though, because those biopsy punches like leave scars. Like
Yeah, I
you've
remember
done
that.
I've done biopsy punch, and they do leave like scars. And it's difficult with these things because like how do you measure like scarness?
Yeah.
Or like a lot of this, or like wound healing, this stuff can be pretty subjective with a giant like delta, right? Where it's like the wound could heal, like when do you decide that it's healed? It's like, and then when you decide it's healed is like, what's the standard deviation, right? Like how many means? It's like, I'm not gonna like do 80, to get like a mean for this shit.
Right.
It's like I want to do at most.
It just looked like,
like more likely like 2, 1 control, 1.
And if you did like my little biopsy punches, I have 6. It's just a little grid.
Yeah.
With a small enough one, I think you could probably do like 10 and get 5 and 5 or something.
Ugh,
Yeah.
maybe. I know I'm the one who's like always weirded out. And then I just like stab myself.
I know, right? You're like, yeah, it's a funny thing about biology, right? Is it's like human biology is really interesting. There's a lot of cool things you can do because we have all these human cells to work with, but getting them is never that fun.
Yeah, I'm really interested in this though, because we do have the stem cells, and now it's like just waiting to get injured.
Yeah,
I really want to see. And there's ways, I have the stuff they use when you have tattoos, like Sanoderm. What
yeah.
it is like you can seal liquid inside,
I thought about
right?
that. Yeah,
And so
yeah.
you could like
Some
pipette a little bit
stuff.
of liquid on the wound, put Sanoderm on top of it, so it holds liquid there with the stem cells in place, and like with media and everything,
Yeah,
and just like let it sit. Like generally, when you have a tattoo, you keep that stuff on for like 3 days. You
yeah,
know,
yeah.
like that would be super
And it
interesting.
would be interesting because you could see it, from the outside clearly. Yeah, that would be interesting. And see, the
Yeah,
the other thing is you could take like a few biopsies, which you guarantee will scar, and then do some side by sides, pretty close together, so it's like the same skin, same tissue, whatever. And then, uh, yeah, so your control would have to be like media plus, the Sanoderm and all of that minus, just the cells.
yeah, yeah. That I mean, yeah. I imagine you could just do like on each leg or on each arm,
Yeah,
something like that.
yeah, it would
Just
be
like right
interesting.
next to each other on the leg or something.
Hmm.
Sanoderm's pretty big, so you can't put them like right next to each other. But like, imagine, yeah, I don't know.
And you wouldn't want to have a situation where you move and it leaks and it spills into the other one, and now your negative control is contaminated with some stem cells. Uh,
Could you use biomaterial that you make as a sort of sand? I mean, the problem is just like keeping it in place. Sanoderm is like just plastic wrap, basically.
yeah,
Yeah, yeah. Control would just be like cells with just media and no stem. I mean, wounds
just
with just media and no stem cells.
yeah,
Yeah, so that
yeah,
would be interesting. That might be. It's like, do I want to wound myself so soon after just wounding myself?
yeah. Mine did eventually bruise, it's like, but it's not
Oh
bad.
yeah, you're bruised.
Yeah,
Dang. Oh,
yeah.
mine didn't. Mine didn't. I wonder if you also did like all those injections with the lidocaine, though. I wonder if that contributed to it.
It could be. It could be. Um,
I'm
but
trying to
yeah,
make you feel bad about the lidocaine. You know.
no, I definitely don't feel bad about the lidocaine. Next time I'm using lidocaine again,
I'm
I didn't feel it.
teasing.
I didn't feel a damn thing. I was very pleased with how the lidocaine came out. You can get FBS for cheaper than $540. It's just really hard. Uh, Collagen sponge laced with stem cells. I've actually heard of some similar stuff being done. I don't remember exactly what it is, but I've heard of some like wound closing biomaterial sort of things.
Oh yeah, that's an interesting idea. Like using like a band-aid or something.
Yeah,
The thing is, you'd have to keep. The thing I like about the Sanoderm is that you have to keep it wet,
yeah,
and then like I think like after like because usually cells take like what um
it is.
like 8 to 24 hours. I mean, sometimes a lot faster, but like let's say to get a lot of cells, like 8 to 24 hours to start attaching.
Yeah, yeah, yeah.
Cool.
Especially if they're not digested, because
Yeah,
I've noticed like if you trypsinize cells, they take a little bit longer. But I guess that's because they have to like regenerate cells. But if you grow them in like suspension
yeah,
culture,
regenerate the
they
protein.
attach super fast because they've got all the surface attaching proteins, just nothing to stick to.
Yeah. Thanks. You like my Pussy Riot shirt? Yeah, that's interesting. Yeah, I wonder if you just instead of trypsinizing, just scrape them off. Then the thing is I don't. Is the thing about scraping or like um, liquid cultures, they tend to clump.
Yeah. Which I
Yeah,
don't know necessarily that would be a terrible thing, but even trypsinized, I mean, still, it's like it adds a few hours, but whatever,
yeah, yeah,
not a huge
yeah. Maybe
deal.
just like trips and digest. Yeah, it probably wouldn't matter too much,
Yeah.
right?
Yeah.
Trips and
Because
digest, wash them a couple times to get rid of all the
yeah, 'cause
trips
the
that are in
Sanoderm's going to keep it there for like days. I mean, normally you put it on a huge ass back tattoo for like 3 days or something, right?
Yeah, yeah. I imagine you wouldn't want to keep it wet for too long. But the media has antibiotics and stuff in it, which is good.
Yeah.
But yeah, I imagine like I don't know, 24 hours or something. I'd want to do maybe.
Yeah. Maybe try different timelines, just do a whole bunch of punches and have like one, you just peel one off each day, see how long. But it would also be interesting because, like, if you could get scarless healing or reduced scarring healing, the interesting thing would be what if you had a pre existing scar and you removed it and then used that? Like, could you get it to heal away a scar that was already there
Oh,
versus,
weird.
yeah, I know.
Maybe. But I have like I have some huge scars,
Yeah,
and like it sounds pretty.
pretty aggressive. Yeah, you have to start with a little scar, right? With
Oh
me.
yeah, like a little tiny one.
Yeah,
Yeah.
I've got a bunch of them, but no, they're all on my hands and stuff or on my legs. But yeah,
Yeah, yeah, that'd be interesting. Because I wonder, like, could you keep? Is there a way that you could just like keep a stock of stem cells alive? You probably could. If you let's say you had a minus 37 incubator and you just kept them in solution, right? Instead of you like put them in like a 15 ml Falcon tube or something. And because usually those last a lot longer than when they adhere to a flask, right? Um, so uh, you just keep these cells in a 15 ml conical for like in your 37 incubator that you keep in your house because it's the future and you have a 37C incubator in your house where you store your stem cells to heal you when you get
yeah,
injured.
your own personal bioreactor.
Yeah. And then like uh or you just have some frozen that you can dump in.
Yeah,
I wonder how well these cells grow in suspension. I haven't really looked into that.
part of me would be interested in putting some sort of a marker in them before doing the healing thing because it would be really weird if it did heal really nice, but then you could also like detect that the stem cells had really like you know
Oh, what kind
had
of marker? Like GFP or something?
I don't know. Maybe. I feel like GFP would be really hard to detect because I don't think they'd be like a majority. Maybe so. I don't know. But I don't know. Even if it was some kind of enzymatic or something you could detect with an ELISA or something, like maybe you have to rebiopsy it.
Fuck that. You're like biopsy it, heal yourself, and then rebiopsy it. Like, oh fuck no.
Yeah. Or I guess you could do, see, like anything that's a protein worries me that it would just, your immune system would kill those cells off because they'll notice weird proteins. But
Yeah,
like,
I mean it might be too hard probably to do anything. Um
I guess you could probably take a
oh,
biopsy,
GFP.
extract DNA, and use PCR to try and detect it.
Yeah,
If
you
you
don't want
added,
to do another biopsy though. That's like defeats the purpose.
well, I mean, you probably wouldn't have to if you use something like PCR. Like if it was on the skin, you could probably just like scrape a few with PCR.
Oh, try to like scrape some of your skin off the top and
Yeah,
see if there's some. Yeah.
yeah, some skin cell
I like
label.
though the idea of GFP, but yeah, I don't know. I don't think a lot of the cells would.
express it enough to
Yeah.
do anything. The nice thing about stem cells is you could do genome engineering instead of just plasmid, and make it a lot more like stable.
Yeah. Man, so many cool things to do with these stem cells. It's pretty exciting.
Yeah,
Opens up a
and I
lot
wonder
of
if you gave them
fun
like a
projects.
yeah. It'd be interesting, especially if you had like a genetic disease, because like I know I've seen things for like people trying to like regenerate lost cell types. You know,
Hmm.
like I think I've seen some diabetes research where they're trying to regenerate damaged insulin producing cells.
Yeah, the beta islet cells or whatever.
Yeah.
Yeah, yeah, that is interesting. But I mean, getting it to your pancreas would be hard.
Yeah, it's not very DIYable.
Yeah, I was thinking about that. Do you like making fat from these cells? And because here's the thing about fat that's really cool is it's like super easy to purify, right?
Yeah,
Because fat just separates from liquid,
yeah,
right? So if you wanted to take stem cells and make fat, um, like you to make the fat, you don't really need anything. And then to use the fat for stuff, that would be super cool to like cook or something with
make,
the fat.
make some human butter.
Yeah, I mean
I mean,
it's human, but like the weird thing is I don't feel like it's like that because it's like it's from stem cells, it's grown synthetically in like a bioreactor and shit like that. So it's like it's less gross to me than like if you were to take your own fat or something like that.
yeah, yeah. Like I took, like, see, like that was after I pipetted off the like after centrifuging my fat. Like obviously a bunch of the fat cells got ruptured during the
Yeah.
process, and so there's just this layer of oil at the top. So it's like the media that you put in, like the blood cells and stem cells at the bottom, a little layer of fat cells, and then just a layer of human oil. And so it's just weird to see just some human oil. But yeah, I mean, it's like, I mean, what is that? Like, if you get, like, is that lard? I don't know what that is.
Yeah, no, it's
Grease?
yeah. I think it's really interesting. It's like a renewable source of um yeah, the cool things, no, we definitely plan. I mean, we're working on it right now. So we already started trying to differentiate our stem cells. So we're
Yeah,
already working on doing some of this stuff.
yeah.
Um,
And that's
we're
the other interesting thing is you could, if you could grow them well, you could grow all kinds of weird, cool things, by turning them into different cells, and then, you know, growing different tissue types.
Yeah, we're just like bullshitting right now and spitballing, because like trying to figure out other stuff we can do with these cells, and we got a bunch of ideas. It's just like, you know,
Yeah,
executing them, right? And so, like, we definitely want to differentiate the cells. We started differentiating the bone. We want to do neural cells and fat cells. I think we have most or all of the stuff for fat, and there's one compound we need for neural cells,
yeah,
but it's pretty expensive. So we'll see. But yeah, it's we plan
yeah.
to
And
do a lot of stuff with these.
I think the bone cells are particularly interesting to me. We talked about this because they can mineralize and make stuff, you know, that just kind of blows my mind to be like, I'm just going to grow some bones, even if it's like little chunks of calcium deposit or whatever. But like, if you were to get really good at it, I don't see a like theoretical reason why you couldn't grow large structures. Like maybe we're 50 years away from that, but like,
You know what you could do though? I wonder because they have a different density, so it'd probably be really easy to centrifuge them using like
they go to the
even
bottom.
like glycerol or phycol or
Yeah,
something like that to like
sucrose,
separate out the
column.
cells
Yeah,
and sucrose gradients are a bitch.
I've only ever used them for sperm.
Oh
And
yeah.
yeah, and it's easy because live sperm swim, dead sperm sink. So
Oh yeah. No. Have you ever done a sucrose gradient where you do like, because they separate, right? So you can do like 20%
Heavier.
sucrose,
Yeah.
30% sucrose, like 40%. You can
You
put it
get
on
the layers
top
between the
of each other.
sucrose layers. Yeah.
Yeah, and it actually, like people actually do this, you know. I had to do it before when I was doing like RNA isolation for something in graduate school, and you just have this like generally what you do is you have like a little dropper that's connected to like a syringe or something, and you just like let the drops fall, and they like it's crazy how they separate though, just like so easily. You think that they'll like bleed into each other, but they really don't. You really get like nice separation of density. And then yeah, don't disturb it, but yeah, you stick in the center,
Yeah. Yeah.
spin stuff down, and like you really get nice like separation because as the heavier stuff obviously.
Yeah. There's some talk, which I don't necessarily believe, about separating sperm based on whether it has X or Y chromosomes, because Y chromosomes are so small that like theoretically a Y chromosome sperm cell should be lighter, but like the weight of DNA is pretty small. It's like the difference in weight between
So DNA base is what, like 100 Daltons? Each gene
yeah,
is about 100 Daltons. So like how many bases are in the X chromosome?
yeah. I'll look
Look
up
this
how many
up.
are in the Y. We'll subtract.
Bases in X chromosome. 155 million.
And the Y has 59 million.
59. Okay, so let's say 100 million. We'll make it easy. So it's 200 million. So it's 2 billion Daltons. 2 billion Daltons to milli. 2 billion Daltons is 3 to the negative 15th milligrams.
So not a lot.
Well, I thought it'd be more.
Yeah, it's yeah. And
I
so
thought I thought it would be like, were we right on that calculation? 2 billion, yeah, because it's like 100 million, 200 million times. Uh, wait, 200
it would just
million.
be there's 100 million per chromosome, so it would only be one because it
No,
would
but
be
it's
X,
no,
Y.
it's 100 million base pairs. Oh, it's
Well, wait,
100.
Daltons
100
to yeah,
Daltons. So wait, 100 million times 100. I think I got that wrong.
yeah, yeah, and it would be even less because yeah, yeah, and it would only be one chromosome because it's going to be XY instead of XX. So yeah, no Daria today. She has something to take care of. She's okay
Sorry,
though.
we're doing all these dumb calculations.
It's just funny to me.
All right, so it actually is 3 times 10 to the negative 11 milligrams.
Okay.
Wait,
So
wait? Okay, so a microgram would be 10 to the negative 3
yeah, you're way
milligrams,
below pico. I don't even know what's below pico.
and then micro nano, that's 10 to the negative 6. Pico is 10 to the negative 9.
Yeah. What's next? I don't even know. Who knows metric?
Yeah, but that's not like. I mean, it's
but
outside
also
the realm of measuring.
yeah, yeah, exactly, and also consider. So you're trying to make like you're trying to separate the cells, but also the force of a sperm swimming is so much greater than that
It's true.
that like the angle of tail has so much bigger of a force than that weight will ever be that it's just garbage.
You'd have to use like an ultra centrifuge with super high density, you know, and that would probably kill the sperm cells or
Everything.
something.
Yeah, for sure.
Femto is negative 15. What's negative 12? Or, yeah. Oh, negative 15 is what we're looking for because
Yeah.
that's negative 12 milligrams. Okay, so it'd be femto. Nanopico femto. Yeah, that's true. Femto. So it'd be like 100 femtograms.
Which almost doesn't exist.
200 femtograms or something like that.
Yeah. Oh man. But yeah, so there's all kinds of weird stuff like that. But you can definitely use centrifuge to separate like dead sperm from live sperm, or like round cells and debris and stuff, especially talking about animal sperm, because it's pretty common to get dirt and shit.
Yeah.
I don't think you can turn sperm cells into anything because they're transcriptionally silent and they don't replicate. They're not really complete cells. They're just like nuclei with.
I don't think that's a question. An actual question.
Oh, I don't know.
Can we turn jizz into stem cells?
I mean, people like to learn stuff. I mean, the benefit is that's a really collectible cell type, right? Like painless cell collection.
Anyway, stem cells. Really cool was that paper that came out with stem cells.
Yeah, where they turned them into embryos. That's kind of crazy.
Yeah, that's pretty crazy. And it didn't seem so. It seemed like they took pluripotent or embryonic stem cells. Then they put one gene. They overexpressed one gene in one set. They overexpressed another gene in another set. Just two different genes, and then they combined all three of these populations: stem cells with no modification, stem cells with modification to make a trophectoderm or something, and stem cells with modification to make an endoderm. And they put them in, they say like they're embryo flat, which is it just had like pressurized oxygen and shit.
And I'm sure there was actually DMEM. And
yeah, no, I looked up the media recipe. It was literally 20% FBS, DMEM, and like 1% pen strep.
Yeah. Okay. This is like nothing.
Yeah, so the media wasn't anything. To grow, the thing is like all the stuff before was the hard part. To keep the population of embryonic stem cells alive, and they said in the paper, it was crazy. They said they tested like 50 different populations of embryonic stem cells or some shit.
Yeah. Yeah, no, even it looked like they brute force the shit out of it too.
Yeah, it
was like 58 in 10,000 or something of the.
There's also like 50 authors on the paper. It's just like, damn. Yeah.
But still, though, like it's kind of crazy. Just take some stem cells, clump them together, then they're like, yeah, we'll become a mouse, and like apparently it had like a beating hearts and shit like that.
Yeah, no, it actually developed. Like it literally developed into like a day. They said it didn't develop past like day 8.5, or they like ended it. I think it just didn't develop past day 8.5 or something. But because why wouldn't you let it grow longer? That doesn't make any sense. But it looked like a normal embryo.
Yeah. Yeah. Other than the fact that like didn't have an umbilical or something, which I think is probably a big part of it's a big limiting factor on how far you can go.
Yeah, but that's pretty crazy. It's pretty interesting because just taking stem cells and be able to do that, and it's just it's wild that they just like self-assemble like that, into these structures. And people have done that with organoids and stuff like that,
but
that's pretty wild.
A whole new level because like you go from organoid to organism, and David should try it now. Okay,
don't
don't
respond to stupid questions.
Well, I didn't read the whole question before I started. So but yeah, so if you took see that's kind of like my like sci-fi vision of the future. Like 60-year-old me sitting there with my like vat that I can grow creatures that I engineer in, right? So like
Yeah.
if like that sounds like the beginning of that technology, right?
Oh, for sure. I mean, how much do you really need it? Who knows? Like, it's really cool, but I'm sure it's a lot easier just to like inject an embryo and do that. But
Sure. Yeah.
it's
Yeah.
it's still, I think, um, from the point of view of maybe it helping create embryos that can easily survive outside the womb because you can grow more tissue types or something, and help them survive. Like,
Yeah, and that's kind of what I want, right? It's like a fully artificial womb machine, you know?
yeah, because I mean that would be epic.
Yeah. And not only just for like all the things it would do for people and even like animals and stuff like that, but also just like all the like science stuff you could do with it because then you could grow shit that like maybe nothing could incubate, you know,
Yeah, yeah,
or nothing alive.
yeah, yeah, and it's also like, yeah, like I just like the hardest part about working with animals is that you have to work with animals.
Yeah, for sure. Yeah. Yeah. And it makes the ethics and stuff a lot more complicated too, right?
Yeah, because it's just like, having mice colonies or something to like engineer mice, like that's the hard part,
Yeah.
you
Yeah.
know?
And like, you don't necessarily realize it if you just read a headline about, oh, they made this GFP mouse or something, but they probably killed like a lot of mice to make that one mouse, you know?
Yeah,
It
for
was like
sure.
multiple rounds of sacrificing just to like collect cells and do other stuff. And like, so this one, they have grown a chicken that lived in vitro. It's like, yeah, but that's totally a cheat because like chickens normally grow in vitro, right? Like,
Yeah, it doesn't make
like an
sense.
egg cell, like if you just like take an egg shell off and replace it with a plastic jar that was breathable, I'm sure it would work just fine, right? So it's like
Yeah, I don't know.
that one's a cheat. Show me a mammal, and now we're talking about something.
Yeah, a mammal that doesn't have eggs.
Yeah, yeah. Skip the platypus too. That's cheating also.
Yeah, yeah, no, I mean chickens are normally grown in vitro. I don't, doesn't
Yeah,
make, it's
yeah.
not
So
that.
that's the kind of thing. Is like like it just sucks that we happen to be a species that like reproduces in like the hardest fucking way. Like if humans laid eggs, like a lot of like gene therapy and all this stuff would be so much easier.
Eggs
You know,
don't grow though, right? So
what do you?
like, what's the biggest animal that comes from an egg?
I think ostrich for sure.
How big are ostrich eggs?
Well, no, no. I mean, alligators get even bigger, right?
Oh, that's
There's
true.
like 18-footer kind of super alligators
Oh,
out
that's true. So you can get pretty big even though if it's from a small egg. I was just
there.
thinking
Well,
like,
also fish. It's got to be like an ocean sunfish then, right? Because they get to like tons, and they come from little bitty eggs.
yeah, I just mean, yeah, that like, I was like, how big would the egg would the humans have to lay?
Oh yeah, oh yeah, I know. Like a human egg couldn't be any bigger than a human baby's head, you know. So I mean, the egg would be like this big maybe, and then the human baby would be like this big.
Yeah,
Yeah,
there would just be a lot more growing to do outside the womb.
yeah, yeah. We'd have to develop very differently, or have like a whole different way to give birth that doesn't involve going through the hips.
Yeah,
But yeah,
yeah, the artificial womb is interesting. It's also like, um, it's interesting for a lot of different reasons because it makes people who aren't normally able to have children, right?
yeah, yeah. Because a lot of people out there have like like their cells are fine, but like their organs are not okay.
Yeah, or just there's something, if you're a gay couple or if you're, You know, somebody who wants to have kids by themselves, or like,
yeah,
it opens up a lot of possibilities.
yeah. That's an interesting thing. I wonder when. So like, I can imagine a future where somebody wants to just like have a kid, but they want their kid to be a clone of themselves. And
Yeah,
like, I don't see why. Like ethically, I don't see a problem, right? I
yeah,
mean, you decide to create a person, whether you partner up with somebody and have a baby, or go get like in vitro fertilization or whatever. If you decide that the baby you want should be an exact duplicate of you, like it's a little different, but I don't see why it's like unethical.
that's an interesting idea because you could totally, do that. You could totally, yeah, create an
Yeah,
embryo from just one person's cells, and
yeah, and like we have the technology. It's just illegal in a bunch of places because everyone's weirded
well,
out.
can we do it? They've done it in mice, haven't they? Where they've turned stem cells into
Sperm cells and stuff.
sperm cells, and because like what you have to do is you take the stem cells and you put them next to the sperm stem cells,
Yeah,
and you take the stem cells and you put them next to ovarian stem cells or something like that, and then they're able to produce eggs and sperm. I think I remember reading that, but I wasn't sure if they did that with humans or just mice yet, or if they were able to do it with humans or
I don't think I've seen it with humans, but I don't know if that's a technical problem or if just nobody's decided to yet,
yeah,
or if I just have missed the news. But even if it's just like somatic cell nuclear transfer, because like even if you like made your own egg and sperm and fused them, you'd still get recombination. So they still wouldn't be genetically identical to you. They would be like a reimagining of you, be the same genes just shuffled around because you could end up with homozygous traits that were heterozygous in you, you know.
that'd be super weird.
Yeah. So like you could
I have a kid
like
who
get
like
black hair and carry for red hair and have a red-headed clone of yourself, right?
I mean, the whole thing would just be really weird.
Yeah. So, like it would have no more DNA than you except for like mutations or whatever,
I
but it
think
would,
it goes back to the need for like a bioreactor. Everybody needs it. And not like a bioreactor, but everybody needs an incubator in their house for storing stem cells and other things like that.
yeah. And the weird thing is like,
Minus 80 and the 37C incubator
yeah.
combo. Like they're connected to each other.
Yeah. Yeah. And you could just use the waste heat from the -80 to heat the incubator. It's even more efficient than having two units.
Who wants to help us make this?
Yeah. We, we need some seed funding.
Everybody needs one in their house. Why? For the future.
Yeah, but yeah, you could just do a clone like somatic cell nuclear transfer, like Dolly the sheep cloning, um, and have your own clone, and that's just your baby. I'm not mad. I don't know why anybody would be mad.
Yeah.
It would be a little weird because then you'd have to go through everything your parents went through. You really get to understand your parents. They're like, oh, I was such a
Yeah.
bad kid. Now I get it. I'm sorry. But
Yeah.
yeah,
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 these stem cells are. I would like to start working with induced pluripotent stem cells. They're a lot more difficult, um,
yeah,
but it seems like an interesting next project.
yeah.
Have we
That's
tried
good.
making some kind of organoid? No, I mean, we are. We're starting to now with our stem cells. We got these stem cells. if you've been following along, and now we're starting to work towards making stuff, differentiating them into stuff. So that's our current goals.
Yeah.
Hashtag goals. Um, yeah, and the goal is to differentiate. You can differentiate into a lot, but differentiate into fat and bone and neural cells. I want to do neural and maybe try to grow neural organoid from that.
It would also be interesting to see like I've seen some papers where they like kind of try to talk to these organoids electrically, and they can like respond in like, kind of coherent ways.
I was thinking about that actually this morning. I was interested in seeing like how well cells grow on like wire, like
Yeah.
actual wires, like a platinum wire or something like that. Like if you just put a platinum wire in a flask on the bottom of it, would the cells grow on top of it?
Yeah.
Yeah. Because I know people grow cells on like like metal surfaces,
Yeah.
like metal pads, and try to. I know there's one YouTuber who tried to grow neurons on these metal surfaces and totally failed.
Yeah. Yeah, but it was like they just bought some store-bought neurons and they died or something. But yeah, I was also thinking like I know there's plenty of like you know, um, like obviously bone can osteointegrate with titanium and stuff. So there's like it might be like you have to kind of explore the metallurgy a little bit to kind of see what the cells are like comfortable with. But like that means proteins can stick to titanium, you know, and they can like build stuff on it. Like I don't know, like if a nerve an ending is looking for something more than just a electrically charged surface, but you know, maybe you could like protein treat the metal first, so it's got like some kind of I don't know receptor.
Yeah,
Something.
a lot of times people will modify the surface of stuff, especially usually generic things like BSA and shit like that.
Yeah,
They
it's
just
like
like,
a compound.
yeah, you just stick it in something so that proteins attach, and then once proteins are attached, that means other stuff can attach to the
Yeah.
surface. Yeah,
So yeah,
attached to proteins. Yeah.
but it would be interesting because like if you had a little organoid and you could like send it electrical signals and then read what it puts back, like even though like I mean you're kind of talking to it, right? You know what I mean, like
Yeah, people have done that for sure.
it would be so
Right?
weird to be like, okay, when I push A, it sends a signal, and then it responds in like a relatively consistent way. It's like, well, depending on what I send to it, it
I think
sends
it's
back.
complicated. That's the
Yeah,
thing with neurons is it's really complicated. My ex partner, she has a PhD in computational neuroscience, and like all they did was try to measure neuronal output in response to stuff, and like that's just complicated. It's
yeah,
not like you imagine it is, where it's like, ooh, look at this thing. I mean, sure, you can cause it to, if you have like some system, you could cause it to like have an action potential or something like that. But if you're actually trying to analyze like the action potentials over time and the response, it's like such a mess that it'd
yeah,
probably be like, interact with it and it has a response, but that response is like unintelligible. Because that's like a whole field of neuroscience in general is trying to decode the response of neurons to input.
right. So what you do then is instead of trying to understand it, you just try and teach it, right? So like, what if you gave it effectively a button it could push that like gives it sugar or something, or, or
Right.
turns the light off? And you just
Yeah.
expose it to some stimulus, and then it like learns to like act, send an output here, and that does some desirable thing. Or even if you just had it like, I don't know, it would be interesting because I know I've seen stuff where they take like a mouse brain, which admittedly this is already like a adult animal brain chunk, and put that chunk on a thing and have it like control something, and they get better over time. And how good they are and how much computation is done in the middle, I don't know, but it's weird to me that just a piece of brain can learn stuff still.
Oh yeah, I don't know. I know they took like a brain, a like human brain, and like grafted it into a mouse brain or something, and they
Whoa.
said that like, the mouse became
Oh,
smarter or
I've seen,
whatever.
yeah, they like put
I mean, it's
human,
like, what does that mean? Became smarter? Who knows? You know, they do
yeah,
stupid stuff with mice. They're like, solve this maze, and they're like, whoa, you're smart.
yeah. And it's like, does it I think it was like glial cells or something like that they transplanted, and they were like, oh, it's, I don't know, they're colonizing and doing something. Yeah.
Yeah, I don't know
So,
specific stuff that they put in the brain, but yeah,
yeah.
yeah, they did do that, but yeah, stick human cells in there. But yeah, the stem cells, a lot of cool things. A lot of cool things to come in the next few weeks. They take time to differentiate and work with, so it'll take time, but follow along. Especially, we're going to start posting more stuff to our website, the-cdc.com. You guys and girls and non-gendered peoples. um,
Folks in general.
Check out the website. We'll post protocols on what we actually did probably by tomorrow, so that you can, get your own stem cells if you want. We're not encouraging anybody to,
Established.
um, try what we tried if you're not trained, because we're
Yeah.
trained medical professionals, in case you haven't known. Actually, I am. I'm a trained phlebotomist, so I'm trained to like stick needles and things.
Yeah.
So like I got that going for me, I guess.
I'm
You
not
know.
trained. I just have lots of practice.
Yeah. How's the parking going to be? To
Good
change
question.
there's no
Yeah.
at the Odin. There's no parking for hundreds of people, especially over the weekend. So no worries.
Yeah. Big old parking lot. Like plenty of things.
But yeah, we've been ranting about stem cells. If you have any questions that aren't weird, or creepy, or extremely off topic, we will try to answer them. So, give us your questions right now if you have any questions.
Yeah.
We'll hang out for a little bit longer.
Yeah. I'm gonna pee real quick because I'm dying.
All right.
Yeah. Hopefully some questions will come through.
I'm just gonna. How about growing teeth? I have no idea about growing teeth. I don't. I mean, growing something like a tooth is really hard and complex. It's pretty big. So, and it's not soft tissue. No idea. There might be some scientific papers out there that you can look up, but I have no idea if you can grow teeth from stem cells. Yeah, stem cells though are pretty exciting. It's great that the technology is available to people. I just wish more people would start using it. Growing human cells, growing your own stem cells from your own body in your own home. It's like, come on, who wouldn't want to do that? And we've been doing tri-lineage assays to check if they're actually MSCs. No, we're doing something way easier. We're just differentiating the cells. How do you determine whether they're MSCs or not, right? That we got from our fat tissue. Well, generally there's two types of cells you're going to get from your fat tissue, right? Fibroblasts or some type of mesenchymal stem cell or something like that. Especially cells that attach, replicate, and culture in the conditions that we provide for them. It's not that complicated. This isn't like something we invented. It's pretty standard procedure that everybody does. So like having to do any complicated assays to like determine if they're MSCs seems pretty excessive. I don't know.
Yeah, especially if you can just turn them into prototypes.
Yeah, I mean, if that's what you mean by tri-lineage assay, which I've never heard of. Yeah, differentiating them is like, we'll let you know. If they differentiate, then like, it doesn't matter. The thing is like, cell types are complicated, and it's something that I'm learning a lot working with cells more and more from animals and humans. And that like, cells aren't very specific, but humans try to be specific about cells. And there's not really easy ways to like determine if a cell is a cell type. Sure, you can be like, oh, let's phalloidin stain this and see if it's like a muscle cell or something. But that's even that in itself is like, you got to fix the sample, kill the cells, right? Whatever. And so you look at pictures of cells, you look at other things people have done, and you start to realize that like everybody is pretty confused about cells, cell types. Even embryonic stem cells, like we talked about earlier, lineages are different. You could have embryonic stem cells, but they function differently than other embryonic stem cells. Even though if you measure that they express, the things that make them embryonic stem cells, they express these things. And so this is all to say, like,
and it's kind of like cell type is kind of a spectrum too, it's like,
it is.
Because all these gene expressions are like various levels and none of them are fixed, right? So like you could have done the same exact measurement protocol an hour later and gotten different results for the same batch of cells, just because like they have, variations in how they express things throughout their growth cycle, throughout the day, throughout whatever. And so like it's all like ranges of stuff. And a lot of it overlaps.
Yeah, I mean, differentiating the cells, that's what we're doing. I mean, if they differentiate into one, I don't think you need to test and see if they differentiate into all of them. Obviously, we want to differentiate them into different cell types, but like, it becomes pretty obvious. That's the thing about this stuff is like, it should be obvious, right? It's like the cells that we got, we don't know for sure if they're stem cells yet, but like what other cell type are they going to be? Especially looking at the morphology and where we got them from,
Yeah, and if they turn out to only be cells that turn into fat and bone and not, whatever else, then they're mesenchymally stem cells. I mean, like, who cares?
Yeah. I think the problem is like science has ruined a lot of things because people try to instead of focusing on the results, right? Like, what are the results we want? Well, we want to turn stuff into bone, we want to turn stuff into neurons, and we want to turn stuff into fat and like play around with those things. Like, does it matter if they're mesenchymal stem cells or not, if they turn into these things? To me, it doesn't, right? Like, I don't care what cell type they are if they turn into these things. Whereas, like, publish a scientific paper, you probably have to do these experiments, make sure they were mesenchymal stem cells, do some gene expression assays to determine if they were mesenchymal stem cells beforehand. And to me, that's like, well, it's not results based, and I'm very big on like applied results based science. It's like if it doesn't work, if they don't differentiate into anything, then we know that they're not mesenchymal stem cells.
Yeah.
If they do differentiate into stuff we want, then we don't care if they're mesenchymal stem cells.
And that's also like assuming that in 5 years we don't decide to redefine what mesenchymal stem cells are. Like maybe they decide that mesenchymal stem cells are a family of 4 different kinds of stem cells that need to be further subdivided based on finer criteria, you know. And
Yeah.
so what we used to call mesenchymal stem cells is now this, and this. And so like shit like that happens all the time. So
Science always gets me with the defining stuff. It's like always trying to define stuff. But not like all. The thing is, we can only define the thing. Like we're not defining all possible things, right?
Yeah.
Because like we don't know all possible things, so we're only defining subsets of things that we experience and know about.
Right.
And then we're using that to create a picture of the world that is incomplete, but we try to make it seem like it's complete because that is all the things we know about.
Right.
And
even though
it's like every year they're like, they found a new organ in the human body.
Right.
Every year there's a scientific article or some article that comes out saying shit like that.
Yeah. And that's the thing is it's like you get these kind of situations where everybody wants to define stuff, but like the universe is rarely as simple as we like to imagine it is, and biology especially is kind of chaotic. And so it's just like,
yeah, biology is especially chaotic. Yeah, again, that's what I learned about cells. When people try to type cells and say these cells are a certain type, it's like, just do it. Just go search on the internet for pictures of mesenchymal stem cells, right? And you'll see pictures of all different things.
Yeah.
And you're like, wait a second, I have a hard time believing those are all of the same cell type, right?
Yeah.
And so something's up. Like either they're not all mesenchymal stem cells, or mesenchymal stem cells is a very broad term to define a lot of different things. And like even cells that I've worked with, like fibroblasts and stuff like that, they can start to get really weird. And like, I don't know if that's because they're just reaching the end of their, life cycle or like what, but they can start getting really weird and turning into really weird cell types. That doesn't look like a fibroblast at all. Like what happened there?
Yeah.
And they replicate, grow still, and you're just like,
yeah. And also it's like, say you have a fibroblast, right? Or you have like a culture of fibroblasts. Like, can you guarantee that none of them are keratinocytes? Like,
yeah.
Or that like
none of them are stem cells.
It's like
usually these cultures aren't pure, Yeah. That's the thing with like most mammalian human cultures is most of them aren't pure. I mean, nowadays, sure, you get like HEK cells. Are those pure? Yeah. But like they're usually not growing up cells, picking a colony, putting it in a flask, and expanding that and being like, ooh, expanding it twice from different colony and just like we have pure cell population here. No, it's like this was just cells that were taken from some human, right? And we isolated. It's like primers or like DNA. Everybody thinks when you order DNA, you get like the exact sequence you ordered, but you don't. You get like 70% of the sequences are the ones you ordered, and the other 30% are just like other shit that they're not willing to clean up.
Yeah. Yeah.
Because everything works out just fine, like nobody cares.
Yeah. 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 I got some longer oligos with like at the cheapest place possible, and it was like, yeah, we can give you this like really cheap price. And I was like, yeah, that'd be great, and I started cloning that shit in, and I got like tested like 5 or 6 different clones and got like a bunch of different shit. There was like various amounts of truncated and whatever. I was like, okay, I should have gone with the more cleaned up stuff. It would have been worth the extra like 12 cents a base pair.
Yeah.
It made so much more work.
Yeah. Yeah. It's yeah, these cell types though, it's been really interesting and funny working with them. Just like, yeah. It's like picking embryonic stem cells. It's funny because it's like the way you isolate embryonic stem cells after you do transformation is by visualizing, right? You can't sit there and like look at the cell and like stain it or something. And like, what does an embryonic stem cell look like? It looks like a fucking weird blobby thing.
Yeah. And then also, like ISPs are the same way. Cause like induced pluripotent stem cells they're like, yeah, you'll know it's induced pluripotent stem cells because they'll have this glossy appearance. Glossy edges is what you're looking for. It's like, okay.
And then you start to realize like how much like, whoa, science.
Yeah. But no, we haven't tried isolating MSCs from any other source. This is our first go at the MSCs.
Yeah, I mean,
other cells, human cells and animal cells.
Yeah, I've grown MSCs before, for sure.
I have.
I've grown MSCs, grown a lot of different tissue types, human and animal. Yeah. This is the first time I've ever isolated them from myself and isolated from fat. I mean, I think the next goal is going to be to isolate some stem cells from blood. I don't know. They're not MSCs, I don't think. They're like.
Yeah, the other ones.
Yeah, they're like some type of cell precursor bullshit.
Yeah, that was one thing I thought was interesting. Is like MSCs can become osteoblasts, but they can't become osteoclasts, which I would imagine in my head them being closely related cells. But apparently, there's a whole different cell lineage to make them that's like more closely related to like blood cells and stuff.
Yeah. 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 Stem cells in general are funny. And like the stuff they use to differentiate them is even more funny. You're just like, what the, like, people just add random stuff and they're like turned into this other thing.
I know, it is really
like
crazy.
Magic potions to differentiate cells.
Yeah. It's like I added, eye of newt and it turned into a whatever cell,
Yeah. Roasted newt, though, is really good. You never had it. Roasted newt.
Never had roasted newt. I think the only reptiles I've eaten have been snakes and alligators.
But yeah, so stem cells. If you want to purify your own stem cells at home, coming soon to a theater near you.
Yeah. Some of the stuff we won't be able to put on like YouTube, just because they will probably.
I think most of the stuff we're not going to be able to put on YouTube. We're going to stop live streaming our experiments and just create videos and put them up on platforms that will host us because we're tired of being banned from social media. And this is kind of a way around it, is to talk about the results in live streams and post videos separately on platforms that allow it. And hopefully, like, they're not like, ooh, you talked about this thing, and so now you're banned. But
well, we'll find out. But I mean, people talk about all kinds of stuff that happened in the past and get away with it. So, yeah. But I had to pull in half my YouTube videos, and you got completely knocked off. And so it's just like that's unsustainable. It's like the whole goal is to like give people access and teach, but then at the same time, it's all getting removed because you're teaching too much and giving people too much access.
Yeah, Robin. I mean, the thing is with protocols and things like that, we want to make them open. So what we're trying to do is create content and like, there's Patreon. We have Patreons. I have Substack. You can see my writings, or Patreon. You can see videos and stuff like that. We're trying to release that stuff behind paywalls because, like, we want people to be able to do this stuff. We don't want people to have to pay to, like, know how to grow their own stem cells. Like, I think that kind of defeats the purpose. So it's like we're stuck in this world where we're like asking people for donations to help us pay to release stuff for free.
Yeah.
Which is like terrible. It sucks. It's annoying, but like it's also, reality. It's like I would rather release stuff for free and not make any money than charge people, because like this knowledge needs to be out there.
Yeah. And yeah, so like the whole point is like the content and stuff, like, like a nice video and stuff, maybe behind a paywall. But as far as like just like a written protocol and stuff like that, yeah, definitely got to just make that stuff open. Otherwise, you're gatekeeping,
Yeah.
Yeah, I think it's interesting. One of the things I definitely want to do is grow the bone cells just for like as long as I can sustain them in a flask, and just try to keep like reseeding it. I just want to see like how much bone can I get in a flask. Like
I'm wondering if you could just like we said, grow it up, centrifuge it down, and just like collect the bone,
and then get them to knit the clumps together.
Yeah, or somehow remineralize it together or something like that. I don't know.
Good idea.
Just glue it together. Maybe you could use meat glue.
I'm sure it's covered in proteins, and it cross-links proteins. But like, I bet you could just like take that as a big clump and put it in a new flask and grow more bone cells. And if they can mineralize it together like they're healing bone, and then maybe make it a solid thing. Yeah. We need like. I'm really interested in the biomaterials aspect of like bone, horn, keratinized tissue, that sort of stuff. It's like, most biomaterial stuff is soft and flexible, and I'm really interested in the idea of like being able to grow something that's like rigid and
for sure,
like has good mechanical properties. Like horn is an impressive material,
It is.
and if you could just grow horn in shapes that you want, that would be super cool.
Yeah, for sure.
And like, but you got to have animal cells to do that. You can't do it with like bacteria or whatever, probably.
Yeah. Anyway, if people don't got any questions, thank you for coming to our live stream.
Yeah, we got work to do today soon.
Yeah, we got work to do. We're also going to put the protocols on our website, so check back on the CDC website, the-cdc.com, and you can see all the work we've been doing and find all the protocols for all the work we've been doing. Check out for new results. We'll be updating, and we'll do our live stream again next week, same time, 11.
Hopefully Dariia will be able to join us.
Yeah, hopefully Dariia will be able to join us. But otherwise, thank you everybody, and we will see you next time.